Hey everybody, welcome back. I'm Dave Cummings from Point Loma Nazarene University, working our way through this paper from Frontiers in Microbiology section by section. And in this episode, I want to talk about the discussion section. When we think of the discussion section of a paper, we've already set the context for the work in the introduction. We've talked in great detail about our experimental design and all those nitty-gritties. And then we got into our results. Hey, look what we found. Now it's time to interpret the results, to make sense of those results. Some papers, the discussion is wrapped up with the results. In other papers, it's separate. The one we're looking at right now, they're separated. So we'll look at it as if they're separate. When they're separate, the idea in the results section is just report what you're seeing. You're not interpreting it yet. When you get to the discussion section, you can interpret it. And then some papers will have another section even after the discussion called conclusions, and that's usually only maybe a paragraph. If there isn't a separate section called conclusions, you can look for a broad, abstract, philosophical, big picture conclusion in the last paragraph typically of your discussion section. So let's zip on down to our discussion section. We get past our results, all our figures and tables, wonderful, fun stuff to read. And then we get to our discussion. It's usually just a few paragraphs, just like the introduction was just a few paragraphs. But remember, the goal here is to kind of contextualize the results. The goal of the intro was to contextualize what they were about to do and why, because there was a gap. And now they want to contextualize now, hey, we've got data. Where does this fit in with what other people are seeing? Does it extend or expand our understanding? Does it feed into what we were hoping to accomplish, the questions we were hoping to ask, problems we were hoping to solve in the introduction? Okay, so keep that in mind as we go through this. So we're thinking about finding meaning in the results, not just, hey, the number was 73, but what does 73 tell you? We're thinking about putting it in relationship, considering it in relationship to other work, and we're thinking about implications. What might this actually mean in perspective or next steps or any glaring needs that are big gaps that we still don't understand or that the new data are begging for us to look into? So in this paper, the first paragraph was just a quick raw summary to set the stage for you. And then the first sentence, and often if it's well written, you can just go first sentence by first sentence through the entire discussion and get a good sense of what was happening. So here they say the four plasmids putatively belong to different incompatibility groups. And then the rest of this long paragraph is all about interpreting what that means. What do we know about Inc-F incompatibility groups, and how might we speculate carefully, very cautiously, about what that means about this plasmid? Now, they haven't actually tested this plasmid to see if it has the same properties as an Inc-F replicon, but there's evidence that it's Inc-F, and all the other studies show that Inc-F replicons behave in this particular way. So there's some cautious speculation in that interpretation. You'll see phrases like may allow, right? So it's speculative. You see something like, thus we might predict. Learn to recognize that when you're reading, that this is not a solid conclusion, that yes, plasmids 1611 and 2011 spread less often and only among enterics, but we might predict that based on the incompatibility groups they appear to belong to. And then there are some, they want to point out something odd here that, okay, I just lost my place. It's noteworthy, that's worth going, hey, something's going to be a little different here. The Inc-R plasmid P3 1611 was captured and maintained initially in P. putida, Pseudomonas, a member of the non-enteric gamma proteobacteria. Although Inc-R plasmids in the literature have only been reported in Enterobacteriaceae, we know that they have the potential, it appears to be, they have the potential to spread to a broader host range than previously thought. This is a real important part of a discussion section where they say, we thought we knew this, but our data suggest it's different. So we thought that these Inc-R plasmids were only found in E. coli and close relatives, and we just found that they're perfectly compatible, this one at least, in Pseudomonas, not related to E. coli, not in that Enterobacteriaceae family, right? Very important types of things to look for where maybe a theory, a model, an expectation is being shaken up a little bit. Next paragraph, they start talking about fluoroquinolone antibiotics because they saw lots of fluoroquinolone resistance in the plasmids. What do we know about fluoroquinolones? And hey, look, we've already done a study on fluoroquinolone resistance in this same environment. So let's do a head-to-head comparison between our old study and this new study and see how our understanding is actually growing and improving. And then same thing, similarly, late generation cephalosporins. And it turns out this CTXM class of resistance genes to these cephalosporins, we've already done a study on that. And we're starting to see them now in the plasmids. So how do our new plasmid studies compare to our older studies on cephalosporin resistance in this same location? Again, we're pushing the boundaries and the frontiers, expanding our knowledge of what's going on in this particular place. And then often you'll see statements like, something greater than previously estimated, right? Again, we thought this, but it looks more like this. So here it says, in the current study, plasmid Petri 2011 harbors a group 1 BLA CTXM55 gene, getting really nitty-gritty here, which was not seen in our earlier PCR-based study, suggesting the diversity of these CTXM genes in the wetlands is even greater than previously estimated. We're building and growing our knowledge and understanding. I like the way this paragraph starts. An unanswered question, right? Again, discussion's a great place to deal with an unanswered question. Let's think about this. You can expect some speculation based on new data, based on old research that's out there. But this remains an unanswered question. And then finally, we get to our last paragraph of the discussion. There's no separate conclusions, so we can expect this last paragraph to be a little more pie in the sky, a little more big picture of, okay, let's get back to the really big picture, and how do our new results speak into our big picture understanding of the problem of antibiotic resistance. So they remind us that chromosomal mutations can arise, but that horizontally transferred resistance genes through conjugation are likely to be even more important. Some people have argued that they've evolved in natural soil and water habitats, and so looking in places like wetlands makes perfect sense because that may be where the raw material is mutating and changing and shuffling and rearranging before it comes into clinically relevant organisms. And then we start to get to their final piece here. The release of antibiotics, antibiotic-resistant bacteria, and antibiotic resistance genes into natural ecosystems. We're in a wetland, right? And it seems to be impacted by this wastewater treatment plant upstream into the natural ecosystems where native bacteria carry their own mobile genome. So you've got this mixture of the fecal bacteria and their plasmids and their chromosomes getting all jumbled up with the wetland bacteria and their plasmids and their chromosomes. It's being predicted here that there's going to be a lot of rearranging and reshuffling, and what emerges may be brand new genetic entities that we need to understand. So the release of all these elements where native bacteria carry their own mobile genome complete with transposons, integrons, insertion sequences, these are the jumping mobile genes, is creating the perfect storm, getting a little speculative, little poetic license there, for the recombination of genetic information into new mobile elements that may find their way back in the clinical environment. A complete understanding of resistance plasmids and the threat they pose to human health must therefore include sustained investigation into the role of the natural environment in the emergence of novel plasmids and thus novel pathogens. Big picture, novel plasmids, novel pathogens. So in the introduction, we started broad, we narrowed it down to here's what we're going to study. Discussion, we said, hey, here's what we just studied, here's what it looks like it means, and hey, let's see how it fits back into the big picture and influences our theories and our models and the ways that we think about the big picture. That's what I want you to get out of the discussion section.